Buenos dias. Good morning, everybody. It's a great pleasure to introduce Dr. Hermes Yeh. Hermes was a graduate student with Donald Woodward in the Department of Cell Biology at UT Southwestern Medical Center, where he started his career studying the neuromodulation of norepinephrine, really a theme that was very novel at that time, but that is carried through with his research really ever since. He then did a postdoc with Donald Puro at the NIH, where he really continued along those lines and really extended his research into second messengers. His first professorship was in the Department of Neurobiology and Anatomy at the University of Rochester. He then went on to Wake Forest, UConn, back to Rochester, and then since 2005, he's been the William Brown Professor and Chair in the Department of Physiology and Neurobiology at the Geisel School of Medicine at Dartmouth. Dr. Yeh has also done a lot of work with the NIH and with the Society for Neuroscience, where he's worked with the departments and programs committee, etc. He will talk today about my soiree with GABA, from phenomena to mechanism of action. Welcome, Hermes.
Thank you. Thank you, Mark. Can everybody hear me okay? All right, great. Thank you for the introduction. I'm really happy to be here. This is not my first time in San Juan, and I've always walked by the Institute of Neurobiology, never set foot in here, so it's great to be here. The house that Jose del Castillo built always has a soft spot in my mind because I actually go through all of the vesicular release papers, quantum release papers with my graduate students, figure by figure, whether they like it or not. So this is sort of a generic title, but before I go on, every time I tell people I'm from Dartmouth in New Hampshire, this is sort of the impression they have of Dartmouth, right? It's a cold place and it snows all the time. And partially it's true, but the reality of it is Dartmouth is actually a very pretty place. You know, after a long winter, we have a very nice spring that comes along, and it's no joke when Thoreau says that April is the cruelest month. And in New England, April is the time when all of the green comes out, all of the roots and plants come out from underneath the ice. Summer is just beautiful, and of course the fall, you probably heard about the fall leaves and things. We're all cluttered with tour buses that come, and is somebody trying to talk to me over there? Okay. Anyway, and this is actually a picture I took from the backyard of my house. So it's a very pretty place, and if ever you have a chance to come up to either New Hampshire or in Vermont, where I live, please let me know in advance, and we'll get something planned. Before I go on, I have to make an obligatory statement here that there's no conflict of interest in any of the content that I will be presenting today, and also that funding for all of my projects have been provided over the years by NINDF, by NIMH, as well as by NIAAA, and these projects really have been funded. And if you notice, either I'm talking about GABA norepinephrine interactions, as Mark mentioned, or GABA modulation in the retina, or by the neurotrophins, but the really common recurring theme in here is GABA. So I named my first dog GABA. In fact, my second dog was GABA too, so I had GABA A and GABA B. And in fact, I thought that I would say something about the modulation of GABA and GABA responses by norepinephrine in the cerebellum today. But then I thought to myself, you know, even though this is one of those where we really moved the story from a phenomenology to mechanisms of action, but with Carlos Jimenez, the adrenergic receptor expert, sitting there, and with Mark being the expert on neuromodulation and neuromodulatory factors, I decided I better not embarrass myself, but stick with something that I know a little bit better. And that has to do with, of course, talking to you about GABA, sort of doing something unorthodox, not as a neurotransmitter that hyperpolarizes neural circuits, but as a developmental neurotransmitter. And I hope that I'll be able to explain to you what it means. And then I'll talk a little bit about that. A lot of this stuff is already published. I'd like to also talk about when this GABA regulation of migration goes awry, such as like when the fetus is exposed in utero to ethanol. Okay, so before I go on, I was just thinking about this with Carlos being the expert and Mark being the expert. It reminds me of a story of Einstein and his driver. Everybody know the story about Einstein and his driver? Okay, but everybody knows Einstein, right? It's a big famous physicist and worked out of Princeton University, right? Came from Germany, never had a driver's license. He hated driving. He rode his bike all the time from his little apartment to his laboratory, physics laboratory, back and forth, back and forth. But of course, he's so famous that everybody wants to invite him to give a seminar in places. He doesn't like cars, he doesn't like trains, he doesn't like airplanes. So he turns down a lot of the West Coast invitations, but East Coast invitations, where he can go by car with somebody driving, he would accept him if he could go. And so he has this old driver who would drive him from Princeton to wherever it is to give a seminar. The driver would go to the back and wait for him to finish his seminar, and then they would go back to Princeton. And this went on for years, many times. So one day we were going to get another one seminars, and Einstein was sitting in the back and he said, driver, I've been giving the same seminars so many times now. Frankly, I'm really sick and tired of giving this seminar. Old driver said, Dr. Einstein, I've been driving sitting in the back room. I listened to you give the same seminar so many times. I have it all memorized in my head. I bet I can just give it like that. So, Einstein sparks start flying between the ears and says, done, right? So they pull out in the rest area, they close, ruffles up his right here, and they move on. And when they got to the seminar place, Einstein sneaks to the back row. The driver gets up here, gives Einstein's seminar verbatim, just a beautiful talk, fills up the whole board, E equals MC squared, and everything. And everything went great. Big applause after the seminar. And then came question and answer time. And this very prominent physicist, obviously a competitor, Einstein, gets up and asks this very, very long question, very complicated. And the audience are all in awe and thinking, what's Dr. Einstein going to say now, right? So the driver just kind of waited very patiently for the physicist to finish the question. And he said then, sir, the answer to your question is actually so straightforward. I bet you even my driver in the back row. This is so it's a good thing that I'm not going to talk about this much. So just to give you a little background, and because I always forget, I'd like to acknowledge some of the past and present.
Could you turn off, mute your computer? Or?
Good morning to those customers on flight 759 with service to Philadelphia. My name is Gabriel, and I'll be working your flight to Philadelphia this morning. Just want to give you guys an update as to why your flight is delayed. Your flight is currently coming from Philadelphia. It is in range. It should be here at the gate in about 20 to 25 minutes. This delay is due to weather, obviously here in Boston and in the surrounding areas. As soon as the aircraft gets here, we will deplane, clean and cater it. And as soon as the crew is ready to board the flight, we will get you guys all boarded up and on your way to Philadelphia. We are showing a departure of about 12:40, but I do believe we'll be getting out of here before that time. Like I said, this delay is due to the weather. If you have any questions, please come see me at 8:34. Thank you. And go up. Yeah, no. Yes.
About 24 hours to 30 hours. And then we looked at these little dots, and here's sort of a blown up image from here. from here. Under control conditions, as you've shown before, as I've shown you before, the cells that go up here in the medial ganglionic eminence will migrate into the cortex. But with the addition of bicuculin, only very few cells will go into the cortex now. And with diazepam, which really juices up the GABA receptor, a whole bunch of cells go. So these kind of experiments alerted us to two things. One thing is that if you just add bicuculin alone into the medium, and it can stop this normal migration in. And if this migration is to be mediated by GABA-A receptor, then that must mean that there is an ambient level of GABA in the slice itself. And that is what's being blocked by bicuculin. And by the same token, diazepam was actually enhancing that ambient level of GABA to augment the migration in. So we did a lot of experiments. I will not go through all of them. We've done a whole bunch of experiments to verify that, in fact, these tangentially migrating cells express GABA-A receptors. We went on to use single-cell gene profiling to actually, and a mixture of that with pharmacology to verify what subunits are available in these GABA-A receptors, and also a whole bunch of other in vitro type of experiments that with which we basically concluded that anything that blocked the GABA-A receptor or anything that enhances the ambient level of GABA, so that the SKF compound and the SNAP compound are inhibitors of the GAT reuptake system. So you add these things. This is for GAT1, this is for GAT2 and GAT3. Bottom line is you add these things in there, ambient level of GABA increases. And if ambient level of GABA increases, that's why you see this sort of increase compared to the controls. And GABA will do the same thing. If you use a GABA antibody to sequester that endogenous GABA level and keep it from working, you see that migration is blocked. And modulators of the GABA-A receptor like diazepam that increases GABA receptor function then increases that migration throughout. And a lot of the stuff we did with the real-time video confocal microscopy because we needed to go in and look at the kinetics, and it turns out what happens is that actually alcohol exposure, addition of alcohol in the medium makes the migration rate faster, not so much the direction, but the rate. So that was sort of an initial set of experiments, and that was all nice. And we kept thinking to ourselves, being interested in the modulation of GABA-A receptors, that if diazepam were to be able to do this in a culture system, can it be done in vivo, right? Can we actually go and affect tangential migration in the fetus that is still in the womb of the mother? But we didn't want to go with diazepam because we wanted to go with something physiologically relevant, right? We're physiologists at heart. And the reason why diazepam was not physiologically relevant is because pregnant women really do not get prescribed Valium or diazepam too much anymore, maybe 40 years ago, but not now. But one of the things that pregnant women, some of them anyway, still do is consume alcohol. And alcohol, or ethanol, is the canonical modulator of the GABA-A receptor. It almost works just like diazepam, right? So we felt that it would be much more clinically relevant and physiologically relevant to actually have a chronic alcohol consumption or a brief binge type alcohol consumption model given to the mom and see what happens to the embryos, the embryonic babies out there. And the model that we used really was one where we had to go back and study the whole process of cortical development of interneurons in the mouse. So there's here's the timeline down here, but just by and large, the GABAergic interneurons that are born in the MGE are born in the MG right around this time, right after the neural tube closure, embryonic 10 or 11, up until about embryonic day 14. And somewhere along the line, after they're born, tangential migration begins. And tangential migration happens at a height. The height of tangential migration happens right between embryonic day 14 and a half to about 16 and a half to 17 and a half. That's about the timeline. And then beyond that, of course, they migrate into the cortex, and then they have to get integrated into the immature cortical circuitry. That can take quite a long time. And then maturation of everything, right? Of their processes, morphology, of the transmitter receptors, and all the transmitter release mechanisms so that they can work in a circuitry. So this can happen all the way up until P60, which is really sort of young adult time already. So if you're really looking at developmental plasticity, it's a very protracted period of time, right? And this is important because later on I'll talk about animals that are exposed in utero, not having seen any alcohol for a long time, but then we look at them again when they're adults. So with this in mind, we set up a model of what we call long-term or chronic alcohol. So we give the pregnant mothers at the time of impregnation, day 0 of gestation, we start feeding them with 2% alcohol. The 2% alcohol is rather mild. What that's going to happen is if you examine the blood alcohol level of these pregnant moms, the blood alcohol level is about 30 mg per dil, 0.03%. Okay? What's the legal limit for drunken driving here in Puerto Rico? Right? So this is equivalent to about 2 glasses of wine a day. Okay? So this is a chronic alcohol model all the way up until the babies are born, but at a low to moderate level of alcohol drinking. Okay? And then we had another model, and the reason why we had another model is because we wanted to get into the mechanism of this GABA effect, just to give an idea. If you remember, this height of migration is right around this time, and we felt that if this alcohol that the mother drinks, which goes readily through the placenta into the fetus, because we can test the amniotic fluid and get 0.03% alcohol as well. If, in fact, the primary effect is on tangential migration, then we should be able to just expose these moms to this period, not this whole period, but just limited to this sort of binge type period of ethanol consumption, and be able to see a similar effect. So that was our second model. Our second model was sort of a three-day binge alcohol paradigm, right smack at this period. But because binge drinking usually means that you drink more alcohol and the blood alcohol levels are higher, our binge drinking model is a 5% ethanol model. And this 5% ethanol model during the binge period gives the pregnant moms a blood alcohol level of 0.08%, just like the legal limit. So we had these two models, and they basically gave the same results. I won't repeat all of them. Most of the results I will give you is from the 5% binge model, because I think that actually is to me, a more interesting and clinically relevant model. Not that the chronic alcohol consumption, the long-term model, is not. 25% of the women, CDC has reports that says 25% of the women who drank before they got pregnant continue to drink throughout their pregnancy. So it is a relevant model. But more importantly, I think this model here really talks about the dangers of early gestational drinking, because this E13.5 to 16.5, where we apply the binge and do a lot of the work, is actually equivalent to the human stages very early on. So here I basically have a caption that basically says that about the third week, okay, in the humans, in about the third week of the first trimester, This is when most women don't even realize that they're pregnant, or when pregnancy tests are sort of false positive. That's equivalent to about day 7 to 11 or lost, right smack at the beginning of the time when we start our binge alcohol. So this is a model basically where a woman who doesn't realize she's pregnant can go to a weekend party, binge drink, and that's it. Okay? And that, I hope to show you, will have long-term effects all the way up until the babies are born and the offspring becomes an adult. So what did we do here? We first, of course, after the binge period, look at the anatomy of we looked at the cells, migration. We looked at the state of tangential migration. Then we let them on go, no more alcohol. Babies don't see alcohol either. Babies are born here. They grow up, and we monitor them. Body weight, everything is all fine. And then by about postnatal day 70 to 80, when they become adults, we then take the offspring that have been exposed to alcohol all the way back here, and we do a bunch of electrophysiology, immunohistochemistry, and behavior tests to look at their neural circuit function that's related to GABA-rich neurons. We look at the disposition of the GABA-rich neurons in the cortex at the time, and we also look at behavior. And we focus on the medial prefrontal cortex. We could have focused on almost any other cortex. But to us, the medial prefrontal cortex was an important part because the medial prefrontal cortex is important in decision-making, executive function. And that is where fetal alcohol spectrum disorder kids have problems with, okay? They have problems with attention mechanisms. And please keep that in mind because I'll come back to that with my behavior stuff. So I don't have to go through all of this. This is simply to say, this is a bunch of numbers and statistics that came from CDC and NIAAA that tells you about the prevalence of pregnant women who self-report alcohol consumption. Now keep in mind this is self-reporting, right? So the numbers are probably higher than this because, you know, the pregnant woman goes to the doctor and sits down and the doctor asks, have you been drinking? What's the first answer, right? Oh, no, I haven't been drinking, right? So, so the numbers are probably lower to the point where the prevalence of basically up to 2 per 1,000 live births is actually higher than what one gets with autism spectrum disorders and attention deficit hyperactivity disorders. And in fact, a lot of the FASD children who go into the clinic are misdiagnosed as having ADHD when really what they have is FASD, okay? So this has actually been very well documented. So the reason why I wanted to bring up this point is not so much to scare people about FASD and autism, but to bring up the point that there's a lot of commonality in the clinical presentation and syndromes of autism, of ADHD and FASD. And all of these, if you remember, are related to interneuronopathy. And therefore, an abnormality in tangential migration of these GABA-rich interneurons may actually be one of the common etiologies to a lot of these neurodevelopmental disorders. Autism, and I realize autism has a genetic disposition to it, and that FASD probably does not. But beyond that, the non-genetic etiology of it is that a lot of these may have things in common. And I think that's an important thing to keep in mind. So what does this binge type 3-day ethanol exposure or ethanol consumption in the mother do to the growing baby? If you look at embryonic day 16 and a half, which is when we stop binge drinking, and this is just a chunk of coronal section of the medial prefrontal cortex that we mark here as the area of interest. So all of our stereology, all of our counts are done in this particular area. And if you really focus on this area and look at cell counts, it's probably obvious here that there are more red dots than here in the control. And very consistent, what we see is embryos that have been exposed to this binge ethanol paradigm have increased cells. These GABAergic palbalbumin expressing interneurons in the medial prefrontal cortex. And later on, in the adult, again, these are adults that were exposed in utero to ethanol, but have not had any alcohol in any other form for 80 days, postnatal at least. Okay, you can see that this increase in palbalbumin expressing, now it's green here, the palbalbumin interneurons actually persists all the way out into the young adulthood. And you can see this, that particularly is obvious in the deeper layers of the medial prefrontal cortex. In fact, what's interesting here is under control conditions, Ewings and others have reported that in fact, deep layer of the pyramidal cells in the medial prefrontal cortex here is really few and far in between. And we see quite a bit more down here. So there is some sorting issue that's going on as well that persists into the adult. And all of these, of course, then gets us to think, again, being physiologists and electrophysiologists, about what the functional consequences might be of this increase in GABAergic interneurons that endure into the adulthood. And when you look at then in the cortex is basically made up 80% of pyramidal neurons and 20% of the GABAergic interneurons. So you might think that a 20% population of GABAergic interneurons may not be that important, because all the rest of them, the pyramidal cells, is much more important. In fact, the GABAergic interneurons occupy a very important functional position within the operations of the circuit of the cortex. The pyramidal cells are basically sorted largely into a superficial layer of layer 2 and 3, and then a deeper layer, layer 5 and 6, down here. They can fire and do their thing, but up in the layer 2-3 region, the firing activity of the pyramidal cells are actually regulated and orchestrated by a group of superficially laying GABAergic interneurons here in red. Okay? And these interneurons are the ones that are going to synchronize, orchestrate the firing up in layer 2-3. And then another type of GABAergic interneuron, this palbalbumin expressing interneurons, would come and actually relay that activity from the superficial layer pyramidal neurons down to the deep layer pyramidal neurons and exert a really powerful inhibitory influence on these pyramidal neurons. And so this then is really what is keeping what we call the excitatory inhibitory balance of cortical function. And that is that GABAergic interneurons play an important role in keeping up this balance of excitation and inhibition. An imbalance of this inhibition excitation is the basis, at least to the best of our knowledge, is the basis of why it is that neurophysiologically we see all of these abnormalities in EEGs, in seizures, and neurodevelopmental diseases like that. And of course, if you have an increased regulation of GABAergic interneurons, you will have an imbalance. An imbalance will favor inhibition. And the bottom line is if you have increased inhibition here, you have decreased output going out from the deep layer pyramidal neurons, because that's how they're controlled. On the other hand, if excitation overrides the inhibition, you'll get an increased output here, and that is simply because the interneuronal integration of the pyramidal activity is not as controlled anymore. So in really checking out and looking at neural circuits in these cortex that have been cortices that have been exposed to fetal alcohol, to alcohol in the fetal stages, we really had this inhibitory and excitatory balance in mind. And so the way to do that was to record from this final output deep layer pyramidal neuron and ask what happens to the inhibitory as well as the excitatory inputs that come in to the neuron. And we looked at that. Again, the hypothesis then is if ethanol exposure is going to increase GABAergic interneurons, then we might expect actually to see an increase in inhibition and therefore an imbalance that favors the inhibition side. And we did a whole bunch of experiments on this thing. This is actually published now in J Neuroscience, so it's not unpublished anymore. But if you just record from cells and look at spontaneous inhibitory synaptic activity, and I say that this is spontaneous inhibitory activity because these were done in the presence of blockers of glutamate receptors, CNQX and APV. So all of the activity that you see here are actually mediated by GABA-A receptor, which can be blocked by, you guessed it, by curcumin, right? And you can see already here that in the adult deep layer pyramidal cells of cortices derived from mice that have been exposed in utero to this binge ethanol paradigm, you can see a lot more of these spontaneous inhibitory events, and this is basically shown here. If you look at amplitude, there's really very little change in amplitude. So increase in frequency, no change in amplitude, usually suggests a problem with presynaptic transmitter readings from the GABAergic interneurons, which is what you would expect because there are more of these now coming in. You can take all of the, this is just some detailed analysis, but you can take all of the little synaptic events that I showed you up here, and you can integrate all of the electrical charges that go in, basically just the area within the curve here, if you spread it out more, and you can summate them and add them up together, and that will give you an idea of the total charge that the pyramidal cell experiences in the case of the binge ethanol exposed condition or the control condition. And you can see that there's a lot of variability, but overall there's a significant increase in the total charge experienced by pyramidal cells in the medial prefrontal cortex of the binge exposed mice. The glutamate here didn't seem to really have too much of a difference. There was a little bit of a trend that lowered, and I'll address that a little bit later, because this really was not an optimal way to look at a glutamate synaptic activity. In addition to spontaneous synaptic activity, you can also look at synaptically evoked activity and get basically similar kind of a response. This is one of those slice preparations in a deep layer where all these big cells here are the deep layer pyramidal cells. The recording pipette is placed here. And we have a multi-barrel pipette system here that we've used, since the day I was a graduate student, I think, where basically 6 barrels are all lined up here. The total diameter here is about 10 microns. So these are tiny little barrels, but they're all separate. And you can put different drugs or whatever it is that you want in there, and focally directly apply each or all the drugs at the same time onto the cell. And this is actually the way we apply bicurculin and APV to be able to block a synaptically evoked response. The very center of this multi-barrel pipette, we put a bipolar stimulating electrode in there. So we can stimulate the surroundings of this cell, get a synaptic response, and once we get the synaptic response, use the different barrels peripherally to apply either bicurculin or blockers like APV and CNQX and see how much of this component is either mediated by the GABA-A receptor, which means that it will be the inhibitory input versus the excitatory input. And here's an example of what I'm talking about. The white line is the synaptically evoked response. This gray line here, or maybe not so white line, whatever you can see from the back, is the response that is left after you apply bicurculin. Which means if you took this white response here and subtracted this response by computer from the white line, you will get this light blue line, and this light blue line should be that bicurculin sensitive GABA mediated response component of this whole thing. And on the other hand, if you were to apply now from a separate pipette CNQX and APV and block out the glutamate receptors, you will get this little response here. And you will get this little response here because this is really the glutamatergic response by subtraction. And this is the larger GABA-mediated response. And I said before, with the spontaneous activity, that it was not optimal to analyze excitatory inputs, and that is exactly the reason why. It's because even at these stages, the majority of the inputs coming in are GABAergic, and therefore pyramidal cell receiving glutamate input from other pyramidal cells are few and far in between. And spontaneous synaptic activity was not going to be able to yield the kind of data we wanted to be able to analyze, and this is the way we did it. And so the bottom line here is, if you look at the GABAergic fraction, again, there's an increase. You look at the glutamatergic fraction, there's sort of a decrease, but not a lot. The bottom line here was if you took the ratio of this and gave us the inhibitory and excitatory ratio, you see an increase in the inhibitory arm of that. All right, so there's an imbalance, and you're actually able to show at the neural circuit level that that imbalance is one that favors inhibition. And of course, again, you look at this and you say, that's all fine, but what about the excitatory arm of it? Well, I won't get into it too much, but there's actually a tiny little bit of a difference in the excitatory part. The bottom line here is the excitatory part does not change all that much. It is the inhibitory part that changes, that shifts that balance towards the inhibition. So we're now, of course, I won't show any data on this, but this is to remind you, to tell you that this project has moved on to really using optogenetics to activate either the pyramidal cell excitatory glutamatergic input or the GABAergic inhibitory input using the AI32 line. The AI32 line basically expresses a Cree-dependent channel relapsin gene, and therefore if you cross it with our NKX2.1 Cree mouse, you get a mouse that has GABAergic and GE-derived interneurons all expressing channel relapsin. And if you're in the cortex, you stimulate record from pyramidal cells, and you stimulate blue light in the NKX2.1 Cree AI32 mouse, you get release of GABA. Obviously, you get a big action potential in pyramidal cells that you record on, but you can also get a postsynaptic current response that is light-activated response that's blocked by QQ. So we're now actually using this as a way of just not looking at tiny little synapses, but looking at how composite interneurons in a field or composite pyramidal neurons in a field can contribute to the overall excitation of the circuits. So switch gears a little bit. I showed you about the anatomy and the electrophysiology. Once upon a time when I was a graduate student, I swore I would never do behavior again because it took so long and I didn't know what to do, and look what happens. It's coming back to haunt me. Of course, the question now is, if you see abnormalities in the medial prefrontal cortex of neuroanatomy, if you see abnormalities in electrophysiology, does it affect the behavior of the mouse, especially behavior that's related to the medial prefrontal cortex, right? So we use a modified Barnes-Smith system. Very simple system. It tests actually two components of it. It tests a spatial hippocampal component and then a attentional medial prefrontal component. So it has 12 holes. The escape hole is usually the hole where we put the home cage or some kind of incentive for them to want to go there. And all the other holes are basically plugged up. And you put the mouse in the center, you test the mouse, and you teach the mouse to go to the escape hole for a 4-day period, each day testing them 4 times. And the mouse first starts out, it doesn't know what to do, it's looking around, spending a lot of time out here, and eventually finds it here. And by about the 2nd day, especially the 3rd day, it gets smarter. You put it in the middle, and it'll probably do this and just go to the hole. And so the latency, the time it takes from here to here, is what you test to see if the mouse remembers. Look at the white line. It takes them a long time first, the first day, second day, third day they get better, and fourth day they get even better. So they learn by the fourth day, they come here, and boom, there's the escape hole, no problem. Then you take the mice, and you give them 2 days worth of vacation. They don't get on the Barnes maze anymore, they don't see it, and after the 2 days, you put them back in here and see if they can remember that they should just go there. And they do. So they have actually learned pretty good, pretty well. And then you play a trick on them. You take this reverse on the escape hole and put it to the opposite side. Okay? And then you put the mouse in the middle that have learned. What do you think the mouse does? Go to the original escape hole because they learn, they remember, right? Very good. Very good. So, but the control mice will go up there a couple of times and say, oh, wait a minute, you're kidding with me here, right? And then they'll discover and they'll eventually learn, actually quicker, to go to the escape hole now, okay? And that is basically here. But look what happens to the ethanol exposed mice. Keep in mind, these mice were exposed in utero for 3 days, and then no more ethanol. They're 80 days old now. And they cannot do this adjustment. And they keep going back to that same quadrant. It's not like they go to other quadrants. They keep going back to that quadrant. So they have learned, but they cannot adjust to a new environment. They cannot make that new decision. That's medial prefrontal cortex. Okay? The fact that they learned all of this is spatial. This is hippocampus. The hippocampus is fine. It's the medial prefrontal cortex that has a problem. And then they eventually will learn. You know what this is like? This is like a chronic alcoholic living in the row houses of Philadelphia or someplace where all the houses look the same. And he's lived in that one house in the middle of that row for 30 years. And every day goes back there drunk, no problem, right? Switch a house on him. And he won't be able, he'll keep going back to the old house. He can't figure it out. And so this is what's happening.
But excuse me, why do the control mice, you can look at it, the control mice are finding it much more quickly than they did
Yes.
during the training.
Yeah, because I think what happens is they get acclimated to the fact that they have to learn this quickly. They just do it. That's exactly right. Yeah, they get better and better at learning this. That's what, actually, that's one of the reasons why we can't do 2 consecutive tests with these mice too. You know, that's exactly why. Okay, good point.
They're as good as they are on day 4.
Sorry.
They are, on the first day of
Yeah.
rehearsal, they're as clever as they were on day 4.
That's right, because day 4 they actually learned. So these mice are actually pretty good. They don't make a lot. They come there maybe once and they'll say, well, this is not right. Boom. Okay? All right. So, and I want to just show you, I sort of showed you the data in reverse. We did these mice behavior first, and all of the electrophysiology and anatomy were done for mice that we tested behaviorally. And that's another important thing. All right?
That's
Sure.
P80. What
Sorry?
stage is that? What developmental stage?
This is, you start at P70. It's a 10-day period, P80. That's why the electrophysiology, everything that you see is P80 or beyond. Okay? All right. So these mice are also more hyperactive. Simple assay. Put them in the middle of an empty box and see if they go to the margin and stay in the center more. And they're more hyperactive. All right? So they're actually more center entries, and they're also faster. This is another one of these clinical syndromes of fetal alcohol spectrum disorder. Hyperactivity. That's why they get diagnosed as ADHD kids. Okay? So actually, I think our model is a very good model to be able to study this. And of course, if we want to study all of this stuff, we want to get to the mechanism of how that happens, right? I just told you a bunch of phenomena. And here, we're looking at mechanism because it's an understanding of mechanism that ultimately is going to get us to that translational arm of being able to contribute to therapeutic and rational design of some treatment. And very quickly, I want to show you some stuff because I think our time is up. But when we get to the mechanisms, one of the things that we thought about, and this is all part of our R21 grant now, GABA, instead of being hyperpolarizing, like in development, it's actually depolarizing during development. Okay? And it's depolarizing because if you look at it, and if you do a voltage current curve, it's depolarizing because the reversal potential of GABA is actually more depolarized than the resting membrane potential. And so when GABA channel opens, the resting membrane potential moves towards the reversal potential of GABA. That means that it moves towards a depolarizing response. Whereas in the adult, it's the other way around. Okay? So it's important that GABA is depolarizing. And the reason why GABA is depolarizing is because, in fact, of the yin-yang effect of two things. An KCC2 chloride cotransporter system and an NKCC1 cotransporter system. These two hold the balance of intracellular chloride concentration. It just so happens that during development, the predominant expression of this chloride cotransporter is the NKCC1, which imports chloride. The KCC2 is almost not expressed at this stage. We've done the immunohistochemistry and Western blot for it. And this is, and therefore, chloride extrusion is much less than chloride import, resulting in an increased chloride concentration and a much more depolarized reversal for chloride. And this then, when GABA receptor channel opens, pushes chloride outward, and chloride being pushed outward depolarizes itself. Whereas in the adult, it's the reverse, and therefore chloride intracellular is lower, GABA A receptor ionophore opens up, chloride flows in, and it hyperpolarizes itself. Okay? This actually happens and during early development and in the mouse during the early postnatal.
Is that why you have inward currents for your patch pump?
Yes.
Oh, that's why I was wondering. I was going to ask why they were.
Well, we could have had outward currents, so it depends on what kind of solution you use for. Yeah. Okay. So the hypothesis that we had was that if, in fact, all this tangential migration is driven by this GABA-mediated depolarization, what if we went and interfered with the NKCC1 cotransporter system and be able to go and suppress this a little bit so that there is actually the chloride drive coming out of the GABA A receptor is minimized. And maybe we can solve this abnormal tangential migration problem. Because if you remember, ethanol acts on the GABA A receptor. It's going to increase the depolarization of that GABA A receptor because it depolarizes at the time. And now if we went and used the drug and blocked or somehow compromised the NKCC1 function, maybe the chloride efflux here would be sort of lower because the function of the NKCC1 would be mitigated. And it just so happens there's an FDA-approved diuretic, actually, bumetanide, that is a blocker of the NKCC1 in the brain. In the loop of Henle, in the kidney, and, I teach this in medical physiology, so it's kind of strange to say NKCC1 because loop of Henle, bumetanide works on NKCC2 and NKCC1. But the reason why in the brain we say that it's selective for NKCC1 is because NKCC2 is not expressed in the brain. That's all. It's not because it's really selective. Carlos.
Yes, it seems to me that you are assuming that because the cell is covalently, that's the reason why it migrates. That is something in the phenotype of the cell that allows them to migrate. That's kind of funny to me in some way.
No. First of all, GABA does not regulate radial migration. We know that. Okay? But that does not mean that it is selective for the migration of GABAergic cells.
So some reason being GABAergic selects them to.
Right. You know, my theory, this will be a great, great thesis project for a graduate student, is that actually GABA, by releasing transmitter as it goes, creates a gradient all by itself to help the migration along. And it's through calcium currents, okay? So this is basically very quickly, I'm sorry, I'm keeping you guys around. But we went back to the organotypic culture system again, and we just basically added bumetanide in there. You know, basically we looked at migration of these TD tomato positive cells with and without bumetanide. And here, for example, you can see here is, yeah. So the white here is the control, all right? So this is the migration cell counts over length and cortex. The details does not matter here, but in slices that have been exposed to ethanol, you get increased migration. You see that. So just bumetanide alone, 20 micromolar, doesn't do much. And in 50 millimolar, same concentration as this, plus 20 micromolar bumetanide, get a suppression of that tangential migration. This is in culture. And you see that in vivo when you give the mom bumetanide and alcohol sort of at the same time, okay? And then this is the blue. I think without getting into too much, you can see that, again, alcohol increases the migration, and this increase in tangential migration is mitigated in the presence of ethanol and bumetanide, okay? These mice, behaviorally, also do much better. Remember this red dot up here? This is the bumetanide-treated group. This is the offspring, okay? So I actually think that there's some hope in this. But of course, again, we have to think physiological relevance. Do you really want to give a diuretic to a pregnant woman, right? And so what happens here is really the NKCC1, by increasing the action of the GABA-A receptor, mediating chloride homeostasis here in the cell, we think is actually activating voltage-gated calcium channels. And it's the calcium channels here that go and interact with the intracellular cytoskeletal elements to increase migration. And therefore, we're targeting now calcium channel, nifedipine as an L-type calcium channel specifically, because you know nifedipine is actually prescribed to pregnant women. It's prescribed to pregnant women to slow down the uterine muscular contraction, right? So actually, it's something that we're looking into. So anyway, the idea here is, of course, that a fetal alcohol spectrum disorder is of embryonic etiology. Then we should go embryonic in attacking it, right? Rather than postnatal. And this is just a hypothesis at this particular point. I hope I've been able to sort of impart to you 3 points. One is that in utero ethanol exposure can affect the tangential migration, and that the effect is a proximal effect because we've been able to show it just by blocking the tangential migration, the time of tangential migration. That interneuronopathy plays a big role in the functional aberrances that we see in the prefrontal cortex of mice that have been exposed to in utero, and treatments and that target either the NKCC1 or the subsequent signal transduction pathways might actually be a promising way to treat fetal alcohol spectrum disorders or other types of disorders that have an embryonic etiology that's related to abnormalities in medial prefrontal cortex. So here's my soiree with GABA. This is GABA here. This is me over here. And it's been happening like this for over 30 years now. But I'm still courting GABA after all these years, I guess. As Paul Simon would say, still crazy after all these years. How many graduate students and postdocs are here? Great, excellent. Excellent. Outstanding. I want to end because I want to dedicate this talk to you for whatever it's worth. This is one of my favorite mentors, Chris Colombre. I was at the Eye Institute at the time as a staff fellow, and Chris was the chief of embryology there, and one of my most best mentors ever. And he just passed away in 2012, like several days before his 90th birthday. But this is something to inspire the graduate students about. That not all easy experiments have been done, but this is to just kind of say, tribute to him that at the behest of his psychology professor when he was working in Jackson Memorial Laboratory, Chris showed motion pictures to a group of dogs. And today, the still unchallenged conclusion of that was that as an audience, dogs are decidedly unattentive. So I thought that was really great. There are still easy experiments to do, trust me. But anyway, Chris also told me that science is a beguiling, demanding, but rewarding mistress. So to all the graduate students, have fun doing it. Thank you very much.
So do the GABAergic neurons that produce their own GABA to affect their own receptors? Is that right?
Yeah.
Yeah. So
they create a gradient.
They're releasing their own GABA, so it's a sort of
yeah.
autocrine.
Autocrine feed forward. Right. That's my thought. I mean, that experiment has never been done. It's not an easy experiment because the way I envisioned it, I talked to one of my graduate students who wanted to do a rotation in my lab. I said, all you got to do is take the slides. We have these cells. They all light up red. All you got to do is do the sniffer pipette experiment, right? Take a little chunk of something that expresses GABA-A receptors, and almost everything expresses GABA-A receptors. Put it on before the growth curve, the bleeding process, and see if you get anything puttering away.
So this should be spontaneous release? Or
yeah, it is spontaneous. Well, it has to be, because these guys they have tiny little sodium channels. They have no action potential. There are certainly no synapses because they're so minute. That's why it's a developmental neurotransmitter. It's not a real neurotransmitter. But anyway, the graduate student ran away and never came back. So must have been a difficult experiment.
So the other thing was, so more of them migrate. So are they missing from somewhere else?
They're not missing from somewhere in the cortex. I think they're missing from the striatum. We see a decrease in the striatum. Whether it's the very same cells that are missing, that are supposed to be in the striatum, that go up there, we don't know. But there is a way to test that. Semaphorin is one of those factors that makes it important. The no-go receptor, whether cells go south into the striatum or go north into the cortex. And maybe by some kind of CRISPR manipulation, you can solve that. But again, that's not an easy experiment.
Yeah, but I guess it was sort of the same line, whether GABA is influencing their decision-making, where to go, or just the speed at which they get to their predetermined track.
I think it's the speed. It's not so much the decision-making, because if you look at the cortex, if it is also decision-making, you would see a differential distribution in density across the cortex. But it's fairly even and random. What it is the rate. There are all kinds of other factors that are the DLX factors, the semaphorin. Gosh, the Caharitzia cells, and I talked to them, that's a whole segment of itself.
So, one thing it looked like, once the number of cells, interneurons you get with alcohol exposure is larger than I would guess the electrophysiological effects you see. I was wondering if you look at the actual synaptic connections that they're making.
Yeah, I tried to get away from that. I was hoping nobody would ask. I actually was hoping that no reviewers would find any of that in my R01. And they didn't. I was glad.
You know who didn't review it.
Your experiments want to call for paired recordings and really look at sort of minimally stimulated response happens. And I think those are really difficult experiments. I mean, I'm one of those who like simple, easy, quick, and dirty experiment. Your experiment is the right experiment to do to look at synaptic strength, but you have to record a pair of synaptically connected cells. And that's not easy. To me, I would rather use optogenetics, shine up the whole slice, and activate all the cells, as many as possible, and look at the composite sort of, activity. I think they can get to the same results.
What about number of contacts? Can you visualize those? Because I mean, there must be some sort of homeostasis that says the pyramidal cells require a certain strength of input, and they're getting too much.
There is data out there, not on the pyramidal cells in the cortex, but Chuck Stevens and others had published a couple of 3 years ago a paper in Neuroscience that says exactly that, right? That pyramidal cells in the hippocampus sort of can only take so much. They have a quota for how much inhibitory and excitatory inputs they can take. I don't know if it applies here. We've never really looked at that. Certainly the pyramidal cells, if they are doing something like that, you would think that the homeostasis would require somehow some kind of retrograde signal that would adjust excitatory input. I haven't seen that.
This is not more prone to be more addictive to other cells, other substances.
Yeah. So here's the addiction expert asking me an addiction. Only thing I can tell you, I run a small lab car, it's giving great. Only thing I can tell you is that if you took these ethanol exposed mice at young adolescent ages, which is about P30, like that, the females drink more. That's all I can tell you. All right? That's something we need to follow, especially nowadays with NIH emphasizing, gender differences, stuff like that. But that's all I can tell you. I basically killed about 3 graduate students already. No, they're all doing very well.
Well, thanks. Speaking of graduate students, I think you meet with them next.